Welcome back friends. Welcome back in another video tutorial for microbiology with Shagni. So welcome back in another video tutorial where we will going to solve some problem from our protein chapter. So this is the first problem where they are telling us that this is a peptide sequence and we have to clip them with trypsin, chymotrypsin, CNBR, that is cyanogen bromide. And they are telling us to find out the number of peptide bonds which will be cleaved, right? So if you have not watched my peptidase video, then you are not able to answer this question, except you have already know about this. And if you don't know about this, then I want to recommend you to please go and watch those videos, okay, so that you can able to understand it. So although I am going to help you here also. So first of all, to solve this kind of problem, first you have to identify whether the enzyme they are mentioning are either exopeptidase or endopeptidase, right? For understanding the detailed concept of exopeptidase and endopeptidase, I again want to recommend you to go and watch the video of peptidase. So trypsin here is the endopeptidase, chymotrypsin here is the endopeptidase, whereas CNBR is a chemical, it is not a endopeptidase or exopeptidase. Okay. Now trypsin, as I have told you in that video, that is it will going to cleave after lysine and arginine, except after lysine and arginine there is no proline, right? So is there any lysine and arginine present? No, that means trypsin is unable to cleave any kind of peptide bond here. Okay, that is why I have mentioned none. Now let's talk about chymotrypsin. So as I have told you, chymotrypsin have the ability to cleave the peptide bond after aromatic amino acids. For example, phenylalanine, tyrosine, tryptophan. And now let's focus here. So here one peptide bond will be cleaved, right? This is this one. Now you may ask, why not this one? So I have to mention that this chymotrypsin is a endopeptidase and it is at the terminal, right? So this peptide bond can be cleaved by only exopeptidase. That is why this is not. That is why chymotrypsin here have the ability to cleave only one peptide bond. Okay. Now CNBR. CNBR I have mentioned that it will cleave after methionine, but here there is no peptide bond after methionine, so it will also cannot. So that is one type of question you may find in your exam from the peptidase. And another type of question they may give you, here they will give you some already cleaved peptide bonds. Sorry, they will give you some already cleaved peptides and you have to find the main polypeptide sequence. And if you remember, we have already done some kind of problems or this kind of problems in case of our recombinant DNA technology, right? Where we have sequenced the entire DNA from fragmented DNAs, right? So those are the restriction endonuclease problems, right? You may also watch this to understand what we will going to do in case of DNA. So now here in case of protein. So here what you have to do, you have to play jigsaw puzzle. Okay, so first of all you have to find out, let's say I have here taken serine methionine. So you have to find out a peptide which have serine methionine, along with that some other sequence. Okay, so this is tryptophan 1 here. So serine methionine glycine threonine lysine. Okay, after that you have to find out something which have at least threonine lysine or at least lysine or at least glycine threonine lysine, and along with that some other amino acids, so that you can move on, right? So here CNBR1 will give you that opportunity, right? Lysine, threonine, lysine, alanine, glutamic acid, right? So after tryptophan 1 there will be CNBR1. Then alanine glutamic acid is also in case of tryptophan number 2. So then tryptophan number 2. Okay, so that's how. Then by combining serine, methionine, glycine, threonine, lysine, alanine, glutamic acid, you will get the entire peptide sequence. Okay, so that is the idea. So, so under such kind of problems, what you have to find out, you have to take any kind of peptide. Here I have taken this one. I may also do this with the help of this fragmented peptides. For example, I will start with alanine glutamic acid. Okay, so I have to find out another peptides that contains alanine glutamic acid. So here alanine glutamic acid present. So alanine glutamic acid, lysine, threonine, glycine, right? Then I have to find out another peptide that at least contains threonine glycine or lysine threonine glycine. Okay, so I may take this one, right? So serine methionine will be added extra. That is how I will get the entire peptide sequence. Okay, now you may ask me that how you will find which is the N terminus and which is the C terminus. Then I will told you the peptide you have obtained, add phenyl isothiocyanate or add your dancyl chloride or add Shanger's reagent so that you can find out which one is your N terminus. Okay, that's how after finding the entire peptide sequence you can also find out which was your N terminus, which was your C terminus. Okay, so that's how the peptide sequencing can be done, and that's how you can solve this kind of problem if you find this kind of problem in your exam. So that is the idea of two kind of problems that you may get from peptide as part of the protein. In the previous video we have already done another problem solving video, that is the charge calculation of proteins. So here another one. So I hope this video will be helpful for you. Thank you.